Hello, everyone, and welcome to today's webinar titled Microbial Air Monitoring Facilitates Disease Surveillance and Outbreak Response. This webinar is part of the 8th Coronavirus Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Sartorius. For more information about Sartorius, go to sartorius.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the left of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You will also notice that this speaker has provided a survey located at the right of the abstract. This survey will be available during the live session and on the on-demand period. If you have any trouble seeing or hearing this presentation, just simply click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located in the abstract window below the presentation window and follow the process to obtain your credits. Without further ado, I now present today's speaker, Clement Arakel, Global Project Manager for Sartorius. For a complete biography of our speaker, please visit the presentation tab from the menu on the left of your screen. Mr. Arakel, you may now begin your presentation, sir.
Thank you very much for the introduction, Sonia, and for having me here. And thanks everyone for joining us today. In today's talk, I will begin with a general introduction on the airborne transmission of pathogens and why it is important to routinely monitor the air in closed public spaces, especially those that are poorly ventilated. I will then give you a brief overview of what we have to offer you: our air samplers, our unique gelatin membrane filters, and the air sampling workflow. I will then cover some trials performed by us and our partners during the COVID-19 pandemic, and also walk you through some case studies from published literature. In doing so, I hope to convince you of the many benefits in implementing a functional biosurveillance program in monitoring and tracking the spread of airborne pathogens. The ongoing COVID-19 pandemic is an unprecedented global phenomenon. We are all familiar with these images of desolate streets during the lockdown, where everything ground to a complete standstill. The pandemic has disrupted lives globally and has negatively impacted global economies since its onset. There have been massive human costs in terms of the lives lost, jobs lost, increased levels of poverty and social unrest. But fortunately, there is hope in sight thanks to the development of several vaccines and new therapies. And there has been a gradual relaxation of restrictions, and the hope is that we all return to a state of normalcy soon, but only time will tell. Apologies if I sound a tad pessimistic. Far from it, to be honest. It's just that we are now confronted with the latest variant of the SARS-CoV-2, the Omicron variant. There is talk of the next hybrid, the Deltacron. Sounds rather ominous and more like a Decepticon from the Transformers franchise. I never thought a day would come when I would groan at the mention of a Greek alphabet. We're all familiar with the Delta variant that wrought havoc in late 2020. We are now facing the currently more transmissible variant, the Omicron variant. And viruses are known to change and evolve as they spread from person to person over time. So what's listed in this slide are only variants of concern. There have been other variants of interest. A variant of interest is classified as a variant of concern when it spreads more easily or causes severe illnesses, or if it has been able to evade the immune response, or if the developed vaccines and treatments have a decreased effectiveness against it. And this is a vicious cycle because an increase in virus spread improves its chances of evolving and the emergence of new variants. So this is why it is essential that we continue to monitor the emergence of new variants by mapping the genetic material of the sampled viruses collected from different sources. And that brings us to the topic of today. There has been a concerted effort to track the evolution of the SARS-CoV-2 virus. So this has mostly been achieved through the sequencing of swab samples collected from an infected person. So there was a strong focus on testing during the peak of the initial waves of the pandemic, but this no longer seems to be the case. It is therefore important that we continue to monitor the emergence of new variants by screening a wider cross-section of the population using other methods rather than wait for an individual to volunteer to be tested. So air monitoring or air sampling is one such method that can be routinely implemented to serve as an early detection or an early warning mechanism. So even though it is now widely accepted that COVID-19 is a respiratory illness caused by the airborne transmission of the SARS-CoV-2 virus and is at the top of the list of airborne diseases, there was a fair bit of debate on the mode of transmission at the onset of the pandemic. The focus was on the transmission of the virus through fomites, or objects that are contaminated through the exposure to infectious agents, rather than the air. So this is why the initial recommendation was to thoroughly wash your hands and regularly disinfect your hands. I assume many of you will remember that besides toilet paper, hand wash and disinfectant solutions were a scarcity. And scientists who were concerned by the lack of recognition of the risk of airborne transmission drafted a letter calling for the recognition of the potential spread of the virus through the air. And paying attention to the call and the growing debate, the WHO drafted a response acknowledging that urgent research was needed to evaluate the spread of SARS-CoV-2 through aerosols. And aerosols being a fine suspension of liquid droplets in the air. So this is why well-fitted masks were mandated as a precautionary measure, and we all now are well accustomed to wearing a mask. And social distancing became the new norm, and we are all aware of maintaining a distance of 1 to 2 meters from each other. So several studies, including this key study published in the New England Journal of Medicine, demonstrated that SARS-CoV-2 in aerosol particles that were smaller than 5 microns could be in suspension in a controlled environment and remain viable for the entire 3 hours of the experiment. So this was quite the game changer, and this raised questions on transmission in closed spaces with poor air exchange. It was gradually acknowledged that COVID-19 could be transmitted either via droplets or aerosols, which can remain suspended in the air for much longer and also travel much further than the droplets. So we are all more than familiar with such infographics. And there has also been an ongoing discussion on how long the coronavirus can remain infectious in aerosols. So a recent preprint, which is still awaiting peer review, was picked up by different news outlets. So this study demonstrates a steep drop in the infectivity of the SARS-CoV-2 virus after 20 minutes of being aerosolized. So this is much lower than the described 3 hours. So this, however, requires further investigation, but is potentially good news in terms of being able to stem the spread of COVID-19. So I'm not going to dwell on these slides too long, but suffice to say that there are a number of examples indicating that airborne transmission is perhaps the main route of transmission. So this is an example of an outbreak in a sports facility. So one of the reasons why you and I couldn't visit our gym during the pandemic. So this is an example of an outbreak in a restaurant. Tracer gas measurements and fluid dynamic simulations predicted that the air circulation could have helped carry the virus over 4.6 meters. And this is another interesting example from a quarantine center in Australia where there were cases of infections in adjacent rooms without anyone directly coming in contact with each other. So it was assumed that some cases of infection were caused by the accumulation of aerosols in the corridor, and others because doors were open simultaneously to pick up meals. So this led to a staggering of meal delivery times to avoid everyone opening their doors simultaneously. So this is anecdotal evidence at best, but an interesting case nonetheless. So the evidence is now overwhelming that airborne transmission via aerosols, especially indoors, has played a dominant role in the COVID-19 pandemic. There are many other documented cases of super spreader events. So choir practice, where 53 members of 61 were infected by one individual, and multiple bus passengers being infected by one individual. And there have been other documented instances of infections spreading where apartments were connected via air ducts. And this brings us back to air monitoring. So monitoring the air for an airborne pathogen is therefore a no-brainer. So there are a number of closed spaces where air circulation or ventilation can be poor. So airplanes, airports, public transport, slaughterhouses, hospitals, ferries, cruises, restaurants, hotels, quarantine centers, classrooms, subways, offices, gyms, cinema halls. The list is long. So air monitoring can therefore serve as an indicator for the effectiveness of ventilation or if any implemented sterilization measures are effective. Interest in air purification systems and sterilization solutions during the pandemic has been immense. Such systems have been an essential pillar in the efforts to reopen public spaces and return to a state of normalcy. However, such installations require qualification, and air monitoring helps establish if there is a decrease in circulating pathogens after its implementation. So we've been approached by several such customers seeking to qualify the air purification or sterilization method, and I will cover one such study towards the end. And by routinely monitoring such public spaces through air sampling, novel variants can be identified, monitored, tracked well in advance to inform risk mitigation strategies, such as implementing health and safety measures and vaccine development or its modification. So a combination of diagnostic testing, air sampling, and vaccination is therefore required to ensure that we have this pandemic well in our rearview mirrors. The focus of this introduction has largely been COVID-19, but there are a number of other airborne illnesses such as tuberculosis, influenza, chickenpox, measles. Any such outbreak can easily be monitored through air sampling. So there were reports of the mysterious COVID-19 associated mucormycosis from 18 different countries in immunocompromised patients. So mucormycosis is typically a rare infection. So the fungus is fairly ubiquitous, being present in soil, but these spores are airborne. As far as I'm aware, it is still unclear what the route of transmission was for COVID-19 associated mucormycosis. So despite being discussed as caused by exposure to mold and it being an opportunistic secondary infection, there were few studies to identify the mode of entry and infection. So a routine air monitoring program in hospitals may have helped trace origins and track the spread of such illnesses that may be potentially airborne. And just to bring this full circle, since we've been mainly discussing COVID-19, the airborne transmission of illnesses is also a cause for concern in poultry farming and in animal husbandry. So these infographics are focused on avian flu and illustrate that an intermediate host is typically required for human infection. However, human infections with bird flu viruses are possible, although rare. So the risks are higher when exposed to a sizable population of infected birds. So the same holds true for swine flu. Flu can spread from humans to pigs as well as from pigs to humans through the air. So this highlights the need for monitoring the air in coops and pens to limit the spread of infection within livestock and to prevent the crossover of viruses from animals and birds to humans. So routine air monitoring can therefore help prevent the crossover of viruses and prevent the spread of an infection through a herd or flock, leading to mass culling events. So now let me switch gears and introduce you to our air monitoring solutions and how it enables the easy sampling of airborne pathogens. Let me begin with gelatin membrane filtration. So our unique gelatin membrane filters have a pore size of 3 micron. But they have the retentive capacities of a HEPA filter, effectively trapping 99.995% of Bacillus subtilis and 99.94% of T3 phages. So it has been documented by other studies that gelatin membrane filters have a retention capacity that exceeds 96%, even for virions that are smaller than 80 nanometers. So the gelatin membrane filters have the retentive capacity of a HEPA filter due to the same principles that governs the trapping of particles within a HEPA filter, such as the sieving effect, an impaction effect, interception, electrostatic attraction, and diffusion. And the filters come in a range of diameters, and they are offered single packed or triple packed and are sterilized by gamma irradiation. The high retentive capacity of the gelatin membrane filter, coupled with the optimal flow rates offered by our portable and static air samplers, facilitates the detection of low counts of airborne microbes and viruses, making this the ideal method for environmental monitoring in cleanroom manufacturing, as well as routine biosurveillance of public spaces. Following filtration through a conventional membrane filter, there may be some concerns regarding the recovery of the trapped microbes and viruses from within the pore structure. So gelatin membrane filters are hygroscopic and are unique in being a water-soluble filter. So recovery is not an issue. So being hygroscopic, the filter therefore also prevents the desiccation of the retained microorganisms and viruses due to the residual moisture content drawn during sampling. So this residual moisture encapsulates the captured microorganisms and viruses and forms a protective capsid, if you will. And this enables long-term continuous monitoring. So we have demonstrated that gelatin membrane filtration facilitates air monitoring for a tested period of up to 8 hours, so without affecting the viability of the recovered microorganisms. I've covered the importance of this need during cleanroom monitoring in pharmaceutical manufacturing in another webinar that can be viewed on demand on our website. Samplers and gelatin membrane filters have been deployed during the pandemic in hospitals, schools, quarantine centers, subways, and other public spaces to monitor the levels of SARS-CoV-2 in the air. So the typical workflow starts with prepping the air samplers, standard cleaning and disinfection procedures. And we offer 2 versions of the air sampler, the MD8 Air Scan that has been designed with cleanroom manufacturing in mind, and the portable MD8 Airport. But many customers of ours have adopted the MD8 Air Scan in semi-static installations. The gelatin membrane filters are mounted on the air samplers and the desired flow rate and the duration set. So I've discussed considerations for efficient air sampling in an interview with LabTube, and the transcript and the on-demand webinar are also available on our website. Once the required volume of air is sampled, the filters are either stored for transport or are processed by dissolving the filters in a required volume of buffer. So following dissolution, the sample can be aliquoted and stored or processed for RNA extraction and RT-PCR. The filters can also be analyzed by a growth-based method by transferring the filters onto a nutrient media plate. And given its hygroscopic nature, the filter dissolves onto the surface of the nutrient agar medium in a matter of minutes. The plates can then be incubated as usual and the colonies counted and identified. Alternatively, the filters can be dissolved in a culture media and the sample used for infectivity studies. We will revisit these workflows in the coming slides. I will now walk you through some of the trials we have performed at Sartorius or in collaboration with our partners to test gelatin membrane filtration as a method for SARS-CoV-2 sampling. So these studies are available as application notes for download from our website, and I believe that some of these have already been circulated to you as handouts. In the first trial, to better understand the detection limit of the method, gelatin membrane filters were dissolved in 1.7 milliliters of 0.2% tween containing 60, 600, 6,000 or 60,000 surrogate virus particles per milliliter. So this is the same as spiking the filters with approximately 100,000, 10,000 or 100,000 surrogate virus particles. I mentioned this because this question has come up before. So the Acuplex surrogate used was a recombinant alpha virus containing the entire SARS-CoV-2 genome. So 140 microliters, or roughly 8% of the dissolved filter, was used for RNA extraction, and 8% or 5 microliters of the resulting eluate was used for RT-PCR. So the eluate was transferred into a TAC-path RT-qPCR master mix that includes two targets for detection of the SARS-CoV-2 N1 and N2 nucleocapsid genes, and an RNase-P target for RNA extraction quality control. So a positive control and a negative control were also included. So I've included the names of the RT-PCR kits used in the following slides as well, should this be of interest to you. And based on the CT values, setting the threshold at 40, we were able to estimate that the presence of between 102 and 1,020 viral particles on a gelatin membrane filter can be detected when what amounts to 1 to 2% of the spiked filter was used. Our partner ALS Malaysia expanded on this trial by spiking filters with 1, 10, 100, 1,000 or 10,000 particles. The surrogate from Zeptometrix contains inactivated or non-infectious SARS-CoV-2. So the spiked filters were dissolved in 2 milliliters of 0.2% tween, and roughly 8% or 140 microliters of this was used for RNA extraction. So roughly 7% of the resulting eluate or extract was used for a quantitative RT-PCR assay. And the Allplex kit detects the envelope protein, the RNA-dependent RNA polymerase, and the nucleocapsid gene. So based on the CT values, we were again able to estimate that the presence of between 100 and 1,000 viral particles on a gelatin membrane filter can be detected when approximately what amounts to 1 to 2% of the spiked filter was used. So when a filter is spiked with 100 particles and only 140 microliters or 7% used for an extraction, the RNA concentration is far too little for a successful amplification. So we therefore recommend that the entire volume of the dissolved filter be processed for RNA extraction to improve the sensitivity of detection. So preferably with a MIDI kit rather than the mini kit that we used. Following air sampling, the filters may have to be stored or transported until they are processed. We tested a range of conditions to determine the optimal storage temperature and period. Gelatin membrane filters were spiked with 10,000 particles and was stored at -80, -24 22 degrees, 37 degrees, 50 degrees, 60 degrees, or 60 degrees at 85% relative humidity from 1 day up to 2 weeks, thereby covering the entire range of storage and shipping conditions. So the filters were processed as before, or roughly 8% or 140 microliters of the dissolved filter was used for RNA extraction, and 8% of the resulting eluate or extract was used for an RT-PCR. To cut a long story short, the surrogate could be reliably detected in almost every case, except when the filters were stored at a relative humidity of 85% for more than 7 days. So the CT values were less than 40, and there was little variation when stored between minus 80 degrees and 60 degrees Celsius. So this demonstrates that gelatin membrane filters can be stored and transported at different temperatures for an extended period of time without affecting the stability of the genetic material of the sample viruses. And building on our initial trials, our partner, New Santix in Indonesia, simulated the aerosolization of viral particles using a surrogate in a controlled environment. So the surrogate was nebulized as 0.3 to 0.5 micron particles in a closed control space, and the MD8 airport was set up at a height of 1.5 meters and a distance of 0.5 meters, 1 meter, 1.5 meter, and 3 meters from the nebulizer. And 3 cubic meters of air was sampled by gelatin membrane filtration, and the RNA extracted from the filters. And the extracts were analyzed using the New Santix MBioCov19 RT-PCR kit. And these kits are capable of detecting 5 genomic copies per reaction. So the RT-PCR kit facilitates the detection of the SARS-CoV-2 helicase and the RNA-dependent RNA polymerase. As anticipated, the CT values were far lower when the sampler was located only 0.5 meters away from the aerosol source. And the CT values for when the sampler was located at 1.5, and 3 meters away from the source were all well below the CT40 threshold, demonstrating that the method reliably delivers on the capture and the detection of SARS-CoV-2 in the air. And the tests were performed on 2 different days, and the outcome remained unchanged. Our partner New Santix then field tested the method in one of Indonesia's largest semi-outdoor mosques. So 3 cubic meters of air was sampled in different areas of the mosque, and the sampler was moved every 5 minutes to cover the space being sampled. And the presence of SARS-CoV-2 was detected in 1 of the 8 rooms that were monitored. It was surprising that the virus was detected given that the spaces were actually quite well ventilated. And this demonstrates that the method is capable of sampling and detecting few viral particles in the air. So New Santix was also approached by a client, Cinema 21, to stress test their air purification system in an effort to woo cinema goers back to movie theaters. So sampling was performed in 5 movie halls. So sampling was performed at the entrance, the cinema halls, and the foyer. Samples were collected before the first screening and after the last show. The filters were analyzed using the sensitive RT-PCR kit from New Santix. So no SARS-CoV-2 was detected in the cinema halls, likely due to the installation of an effective HEPA filtered ventilation system. So this system has been described more in detail in a video on our website. And closed kindergartens and schools with access to only remote learning during the pandemic have had a significant impact on the development of children during the pandemic. Following their trials, New Santix worked with the Ministry of Education to implement similar screening and air monitoring programs in daycare facilities and kindergartens across Indonesia in an effort to reopen the schools and reassure anxious parents that staff and children were returning to safe spaces. And following the preliminary trials performed by ALS Malaysia, they also field tested the MD8 airport and gelatin membrane filters in a hospital and quarantine center. So 1 to 2 cubic meters of air was collected from different zones in these facilities. And the filter was dissolved in nucleus-free water and the RNA extracted from the entire filter. So SARS-CoV-2 was detected in some of these zones. But some of you may have already noticed that in some cases, only one of 3 genes were amplified. Based on our initial results, we conclude that there were perhaps far fewer than 100 viral particles per gelatin membrane filter, with roughly 8% of an eluate being used per PCR reaction. That would amount to roughly 8 genomic copies or equivalents per reaction. And this is in the event of an extremely efficient RNA extraction. And that is a tall order. In reality, the number of genomic equivalents may be fewer than 8 per reaction, and perhaps at the detection threshold of the RT-PCR kits that were used, explaining this variability that we observed. And that brings me to the limit of detection and whether this sensitivity is reasonable in the context of the infectious dose. So how many particles of SARS-CoV-2 constitutes an infectious dose? So there's been some research on this, and the estimates cover a wide range. On the lower end of the spectrum, there are estimates that only 10 airborne particles constitute an infectious dose. Other estimates are between 100 and 700, while others peg this at less than 1,000. So as one can imagine, an accurate measure is difficult since this can vary between variants. So the jury is still out. Our results and those of our partners demonstrate that SARS-CoV-2 can successfully be detected when at least 100 particles are captured per gelatin membrane filter, which is within the presumed range of the infectious dose. So scoring a positive detection would indicate that risk mitigation strategies need to be implemented in the area being monitored. And a method that is far too sensitive or prone to false negatives may also be counterproductive. And you don't have to only take our word for it that the method reliably delivers in the detection of airborne pathogens. In the time remaining, I would like to walk you through a couple of published studies that have successfully been using gelatin membrane filtration for virus sampling. In the early pandemic, a study published in Nature investigated the potential for SARS-CoV-2 to spread through aerosols in 2 hospitals in Wuhan. So the study tracked 3 types of aerosol samples collected in these hospitals: total suspended particles, size segregated samples, and deposition samples. So 30 aerosol samples were collected by active air sampling to study the total suspended particles. 3 size bin samples were collected to study the distribution of SARS-CoV-2 in differently sized aerosols. And 2 deposition samples by passive settling were collected using gelatin filters in holders placed on the floor over 7 days. So following sampling, the filters were dissolved in deionized water and trizol was added to inactivate SARS-CoV-2 and the RNA extracted. The sensitive droplet digital PCR was used to detect the presence of SARS-CoV-2. SARS-CoV-2 was detected in several areas of the hospital, but the concentrations were higher in poorly ventilated areas, such as toilets and areas seeing increased foot traffic. And SARS-CoV-2 was also detected in aerosols in a range of sizes. The researchers also discussed the possibility for resuspension of particles from protective clothing and the floor of the hospital based on the deposition samples collected. So based on these results, the researchers were able to recommend risk mitigation strategies such as improving ventilation and sterilization regimes, wearing of masks in crowded areas, and that the protective gear be sanitized prior to them being taken off to prevent pathogens being swept up into the air. Another study in Milan collected air samples from areas designated as either contaminated or semi-contaminated. And like other studies, the MD8 airport sampler was used at a height of 1.5 meters and 2 cubic meters of air was sampled. And a total of 42 air samples were collected and the results were mapped onto the floor plan of the hospital. And the map paints quite an interesting picture. Air sampling demonstrated that areas housing patients tested positive for the presence of SARS-CoV-2, and areas occupied by staff were negative. And the air samples were complemented by swab samples of surfaces. Interestingly, despite the undressing room testing negative for SARS-CoV-2 in the air, the virus was detected on the door handles and medical equipment in the undressing room. And this highlights the importance of segregating the dressing from the undressing zone. So based on this, the researchers discussed the importance of ventilation, air purification, disinfection, and the containment of patient facilities. The authors conclude their paper by highlighting the fact that a positive detection was only a marker of virus shedding, but it does not necessarily indicate the presence of viable viruses in the air, and that a viral culture should be performed to demonstrate viability. So this is a topic I will revisit in the following slides. And by being a uniquely water-soluble filter, near complete recovery of viable viruses can be assured where required. On a similar note, another team of researchers in Toledo collected air samples in households with patients under home quarantine, and both pre and post air purification samples were collected. 1 cubic meter of air was collected, and the MD8 airport was positioned 1.5 meters above the ground and was located 2 meters away from rooms housing patients. So following an initial round of sampling on the very same day, a Dyson portable air purification unit with a HEPA filter was placed at the center of the room and operated for 1 hour before air samples were collected again using the MD8 airport. And the gel membrane filters were then transported to the laboratory and stored at minus 80 degrees until they were processed. And the filters were dissolved in water and the RNA extracted from the filters for RT-PCR analysis. And following air purification, SARS-CoV-2 was no longer detected in all but one room. And the researchers pointed out that this was likely due to the fact that the room size was nearly 2 to 3 times larger than the other rooms and was outside the specified capacity of the air purification unit that was used. And this study endorsed the use of air purification units to protect other inhabitants of a house under quarantine and highlights the importance of qualifying air purification units through air monitoring. So there are several other studies using gelatin filters. I have focused on but a few. Our samplers have also been widely used by a number of governmental organizations, such as the Chinese Center for Disease Control and Prevention, who have conducted several biosurveillance studies in public spaces. But it is not only during the COVID-19 pandemic where we have seen an increased interest in our air monitoring solutions. And gelatin membrane filters have also been used during the MERS outbreak. So similar air sampling studies to the ones discussed were performed in 2 hospitals in South Korea treating MERS-CoV patients. So this time around, the MD8 air scan was used to filter 1 cubic meter of air through a gelatin membrane filter. And the filters were then dissolved in viral transport medium. And then they were stored at minus 80 degrees until they were analyzed. And the samples were filtered using a syringe filter to minimize secondary bacterial and fungal contamination. And the filtrate was then incubated with Vero cells. After 14 days, the cells were harvested and MERS-CoV detected by RT-PCR. Infected cells were also used for immunofluorescence assays. And importantly, viable MERS-CoV was detected in the air of a room housing an infected patient. So rather than extract RNA directly from the gelatin filters after air sampling, a clarified extract was used to demonstrate the presence of viable infectious viruses in the air. In yet another study to examine whether MERS-CoV could be detected or transmitted through the air, samples were collected in a camel barn owned by an infected patient over consecutive days. So 1 cubic meter of air was sampled using an MD8 air scan. The filters were then dissolved in viral transport medium and stored as before at minus 80 degrees until they were analyzed. So RNA extraction followed by RT-PCR confirmed the presence of MERS-CoV in the samples. There's also been interest in the airborne transmission of pathogens in poultry farming. So this control study generated aerosols containing the infectious bursal disease virus that causes Gumboro disease in chickens and turkeys. The gelatin membrane filtration method was used as the reference in comparing other air sampling methods. In this case, the filters were dissolved in Hanks balanced salt solution medium, or HBSS. It was demonstrated in this study that viable IBDV viruses could be recovered from the filters. It is key to understand airborne transmission of infectious viruses to prevent unnecessary culling of livestock, and more air monitoring of airborne illnesses are required to implement pragmatic risk mitigation strategies. So on a similar note, gelatin membrane filtration has also been used to detect the presence of Campylobacter in poultry farms. So Campylobacter are prevalent in poultry and cattle. Campylobacteriosis is caused by Campylobacter, and the main route of transmission is foodborne via contaminated meat or milk. So prevention is therefore based on stringent controls implemented along the food chain. One of these measures includes ensuring that birds on the farm are kept in a hygienic environment and regular checks are carried out to detect the presence of Campylobacter. So this study compared boot swabs, the traditional method of Campylobacter detection, with samples collected by air filtration. So samples were collected from 44 flocks in 5 countries. And based on the results, the method was proposed as a screening method that is 4 times faster and is 4 times more sensitive than the traditional boot swab method. A video from the team describing the entire workflow is available on our website as well. So I hope I've convinced you of the many merits of the system. Gelatin membrane filtration has been on the market for many decades. It has been widely adopted in pharmaceutical manufacturing industry to qualify and monitor clean rooms across the globe. And our technology has been adopted for the environmental monitoring of advanced aseptic processing systems such as RABs and isolators. And these are some examples of installations in pharmaceutical manufacturing. So given that the gelatin membrane filters are hygroscopic, they prevent the desiccation of the retained microorganisms due to the residual moisture drawn during sampling. And we have demonstrated that gelatin membrane filtration facilitates continuous monitoring for a period of 8 hours, which is typically the duration of an entire manufacturing shift. So the continuous monitoring facilitates intervention-free air monitoring of clean rooms, and in doing so, limits the risk of introducing secondary contamination with every entry and exit. So this is now a key requirement of the EU GMP Annex 1 draft. And as already mentioned, you can learn more on this from the on-demand webinar on our website. Although currently not a requirement since gelatin membrane filters are capable of monitoring airborne viruses, clean rooms can be monitored for any airborne contaminants or pathogens with one single method. And our air monitoring solutions also caters to offbeat applications. Our MD8 air samplers have been used by NASA to conduct microbiological tests on board the International Space Station to gauge sources of contamination. So we've also recently had researchers use our solutions to investigate microplastic contamination. Others have used this to study the spread of allergens, and another for forensic analysis of crime scenes. So I will hopefully get to cover these in perhaps a follow-up webinar as and when the samples are analyzed and the results published. And that brings me to the end of the presentation. Today I have touched upon the many benefits of gelatin membrane filtration for virus sampling. I've also walked you through tests that we and our partners have performed. So I've also covered some case studies highlighting the use of our solutions for pathogen sampling to limit the spread of airborne infectious diseases. And we offer both a portable sampler and samplers that can be installed for static sampling purposes, although many of our customers have used these for semi-static sampling as well. So the methods have been tried and tested for the sampling of pathogens in several environments such as hospitals, schools, public transport, cinema halls, and many other public spaces, as well as environmental monitoring in cleanroom manufacturing. So the filters facilitate near complete retention of microbes and viruses, and recovery is easy given that the filters are water soluble. So this makes the method compatible with both traditional growth-based methods, where the filter can be transferred onto nutrient media, or a rapid method, where the filters are dissolved in a buffer of choice and then analyzed by PCR. So since the viability of the trapped pathogens remains unaffected, following dissolution, the filters can be used for infectivity studies or perhaps the dissolved sample split between different methods. And the samples can also be stored and transported at a range of temperatures from -80 degrees to 60 degrees Celsius for a tested period of 2 weeks for nucleic acid-based detection. So in a nutshell, air monitoring in public spaces is key to identify newly emerging variants and track their spread. And before I wrap up, I would like to remind you once again that these application notes, white papers, and many other resources can be accessed through our website. Please stop by. Thank you very much for having joined us today and for your attention. You have my email address here. Please feel free to reach out should you want to get in touch. I'm happy to take any questions you may have. Hopefully I have answers to all of them. Thanks again, and back to you, Sonia.
I want to thank you, Eric, for your time today and for your important research. And before we go, I want to thank our audience for joining us today. I'd also like to thank our sponsor, Sartorius, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 19th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.